Hello students, welcome to the second last lecture of this course. So, because I am stuck in a conference here at IIT Delhi, I had to record these two lectures. I couldn't take live classes, but I request you to kindly go through them. Okay, have a look at these two lectures and at a later date, like before the final quiz, like I'll organize a doubt clearing session, and that doubt clearing session won't be just limited to these two lectures, but on all the topics that we have discussed so far during this course. Okay, so that while you're studying the topics, even if you stuck, like you feel stuck anywhere, you can freely ask me on the doubt clearing session. So I'll definitely try my best to arrange that doubt clearing session before your final quiz. And also I'll let you know the date and the time for that quiz. So, in today's session, we will continue with the topic, the host response to biomaterials. So, the previous session, we discussed the immune system, right? A brief introduction to what is immunology, okay, or what part of immunology is relevant to us. So, we are mostly interested on how the immune cells behave, like the macrophages or other leukocytes. And then we discuss uh, the effect that the biomaterial have on the host. Like there can be local effects as well as systemic effects. So at the local site, there can be like uh, some of the aspects we didn't discuss very in depth in the previous class because in today's class we will go through them, but mostly we focused on the systemic effects, how the biomaterial affects the host in the overall picture. Like it can cause thrombosis or it can introduce an infection at the tissue site. In animal models, people have also observed the formation of new tumors at the implant site. So these things we already covered. In today's session, we will focus on the local interaction, the biomaterial and the host tissue interaction. Okay, at the site of implant, that is the local interaction. And we will also realize that these are the effects that the host tissue has on the biomaterial. Okay, as we will go through each of these events one by one. So we can distribute the entire biomaterial and tissue interaction into these separate seven events. The first is the injury. So when you're implanting, you're injuring the site. Then due to injury, the leakage of blood vessels and the release of blood at the site will happen. So that is blood and material interaction will take place. Then there will be formation of provisional matrix at the site or over the implant. Then the inflammatory cells will come and do their job. So they can either cause acute inflammation or a chronic inflammation can also result. Then ultimately, a granulation tissue, a pink color, slightly pink color tissue. If you remember, we already discussed this when we were talking about the wound healing process, the normal wound healing process. There also granulation tissue gets formed. So here also granulation tissue will get formed, and then depending on the size of your implant, like if it is a very big implant, then there will be frustrated phagocytosis by the macrophages, and thus we see a foreign body reaction at the site of implantation. And ultimately, the body will try to like isolate this biomaterial, that is fibrosis, okay? Or fibrous capsule development will take place. That is the end step. So while placing a biomaterial in vivo, either through injection, insertion, or through surgical implant, we will have to injure the tissue, right? So the placement procedure, it initiates a response to injury by tissue, organ, and mechanisms, and they are activated to maintain homeostasis. Means what I'm trying to say is when you're implanting, okay, your biomaterial, you are going to disturb the homeostasis of that tissue or organ. So, to what degree are we disturbing that homeostasis, that thing, and also while implanting, to what extent we are creating a pathophysiological condition at that site. So these two parameters will help us decide whether our biomaterial is actually or whether our implant is biocompatible or not. It's basically a measure of biocompatibility of our implant. So if the homeostasis is very much disturbed, that basically it's a scale, no balance scale. If it is too tilted towards the disturbed side, then it will take a very long time for the tissue to regain its homeostasis status. Whereas while implanting, if the normal physiology of the tissue or organ gets very much disturbed and to such an extent that a diseased condition starts to develop. So if that happens, then we are also farther away from the biocompatibility. Means our implant is not compatible to the host. Okay. Now, when we are causing an injury, then the immediate response, what we will expect is the inflammation. So inflammation is generally defined as the reaction of vascularized living tissue to local injury. So injury happened, then inflammation will start. So the purpose of inflammation is to contain, one second. So sorry for the disturbance. So as I was saying, the purpose of inflammation is to contain, neutralize, and dilute or wall off the injurious agent or process. So whatever process that has caused damage to the homeostatic status of our tissue, that is uh, that the body wants to contain, okay, so that it doesn't spread, or it want to neutralize it. If and it's good if suppose a pathogen agent has entered, then the first thing that is like seems logical is to neutralize it, then dilute it. If some drug, very high concentration drug has been injected, then our body will try to remove it from the site, right? And like lower its concentration, wall off. So again, it seems appropriate to say that if any pathogen agent has entered, then it should be contained to that particular site. Or if, say, cancer, if our body detects that a cancer has started to form, although in many cases cancer goes undetected, but in some cases body do try to detect, okay, because it starts secreting some proteins which are foreign to the body and some means which are antigenic, so it can detect. But so in those cases, our body will try to contain it to that particular area. Okay, so. And these processes, which I just explained, what means what happens here, the process that happens during inflammation, they will be followed by the healing process, okay, or regeneration process at the implant site. Now, immediately after injury, what happens? So, due to injury, you will be uh, breaking off the continuous blood vessels, right? You'll be cutting them. So because of which the endothelial cells they get activated, and what they will do, they will change the flow rate of the blood, Then the diameter of the blood gets varied, as well as the blood vessel, okay, the blood vessel, the endothelial wall, it becomes more permeable, or you can say it becomes leaky, so that the inner content of the blood, basically blood or the cells in the blood, they can come out and arrive at the tissue site. Now, the fluid proteins and the blood cells, they will try to escape from the fluid system, just what I mentioned. And this process is called exudation. Okay. So during exudation, or in the process of exudation, due to increased permeability of endothelial wall, uh, the blood fluid, means the serum or, and containing, it has serum proteins, then the blood cells, like leukocytes, especially I'm referring to leukocytes, so they will come at the site of injury. Now, following changes in vascular system, due to arrival of these leukocytes, etc., cellular events will start, and that event will characterize the, or you can basically measure that the inflammation has started. Now, early response to injury, it mainly involves blood and vasculature regardless of tissue type. So it doesn't matter what is the tissue. So this is also true for the cartilage. You know, cartilage is a lymphatic, a vascular, okay? But still, if some injury to cartilage, okay, it happens, so some blood interaction, some blood material interaction is expected, okay? That is why it is written regardless of tissue type. So, when blood interacts with the implant, then the proteins which are in the blood, basically what we refer to as serum proteins, so they will deposit on the biomaterial surface. And from a wound healing perspective, this deposition of protein on the biomaterial surface creates a provisional matrix. And this provisional matrix, it happens, its formation happens within like minutes or hours, like immediately after the implant has been placed at the site. Okay. So what this provisional matrix consists of? It consists of the fibrin. It's a fibrin gel, hydrogel, okay? And it comes from a precursor form, fibrinogen. Fibrinogen converts to fibrin in presence of an enzyme that is called thrombin. I think so. Just check, okay? I think it's thrombin. And then the inflammatory products like the activated platelets, then inflammatory cells and endothelial cells. So these cell types are also found on the provisional matrix. So the provisional matrix, they have these components. Okay. So these components, basically I'm referring to the cells which has the ability to secrete and release various kind of, say, signaling molecules. So these signaling molecules will be helpful in initiating reorganization and repair process. Okay? So you can now understand why provisional matrix is very important. So ultimately, what we conclude that the provisional matrix, it provides both structural as well as biochemical component to the process of wound healing. So as soon as the provisional matrix form, it basically lays down the foundation for the other steps of wound healing to happen. So it's a complex 3D structure, and it is composed of the base fibrin, okay, and within it there will be cells, and that uh, like so how the cells are adhering on fibrin network? They're adhering because the fibrin contains the cell adhesive motifs. Now these cells, they can secrete various kind of signaling molecules like the mitogens, chemoattractants, cytokines, and growth factors. So they will create their concentration. There will be some concentration gradient of these factors or these chemical molecules within the provisional matrix that will further uh, like lay the foundation for various cellular proliferative and synthetic processes. A provisional matrix, they may be viewed as a, if you try to view them in the perspective of tissue engineer, so it can be you can make a provisional matrix if you just simply use a biomaterial which is naturally derived, it is biodegradable, and it allows sustained release of these signaling molecules. Then you will have a provisional matrix. So just a simple way of explaining the nature of this matrix in terms of tissue engineering. Now, due to the presence or due to the structure of this matrix, it allows sustained release. So, it is sustained release of chemical molecules that will allow the molecules to diffuse from the site at a much slower rate. Okay, and thus a concentration gradient, an observable concentration gradient can be uh, created, and that lasts for a significant amount of time. So in that time, we can expect this cell migration to happen and ultimately reach its particular site. If there is a burst release, like instantly there is a high concentration and then the low concentration starts forming, and that too very in a very small time span, then what will happen? Less number of cells will arrive. So we don't want burst release. We expect or we want a sustained release because then only this provision matrix will allow migration of the cells and then leukocytes, etc., at the site of injury and thus initiate the process of wound healing. Although we have gained a lot of understanding of the provisional matrix and what it is capable of, but we cannot really control its formation. Neither we can control its effect on the subsequent wound healing events. This is because most of our knowledge has been gained from in vitro studies. We cannot, we know very little what happens in vivo, okay, or what regulates the formation of the provisional matrix at the biomaterial and implant interface in vivo. But people have like proposed various kind of hypotheses on this particular topic. Now, coming to the next step. So injury happened, all right? So because of the injury, there will be blood and material interaction. And then, due to the deposition of the blood proteins, the cells will start adhering. They will secrete fibrin glue, etc. Fibrin, so another word for it is fibrin glue, which clinicians use. So just it came out of my mouth suddenly. But for you, it should be called fibrin hydrogel. So that gets formed, and within it, various kind of cells are there, and that secretes various kind of signaling molecules that is going to help the subsequent inflammation process. So now we have come to the next step, that is inflammation. Now, the predominant cell type, which is present in the inflammatory response, it varies with the age of inflammation. So, as you track the inflammation process happening, you can also track the cell number of different cell populations. You will see, I will show you a graph where peaks can be observed for different cell type at different time point. Okay. For neutrophils at the beginning, they are the cell which is present in abundant, okay, and the beginning of or at the uh, time when the injury happened. Okay, so they are the predominant cell type in that case. Why? Why this is so? This is because these neutrophils, they are short-lived. Okay, so they die very quickly. So what is the time? They die within 48 hours. And the e-migration of these neutrophils from the blood vessels to the tissue happens in a very short duration as well. So they migrate fast. And the cumulative factors, that is the chemokines, etc., secreted by the neutrophils, or sorry, I meant to say is the factors needed for the neutrophil migration, they are also released at a very early stage in the inflammatory process. So, because of these three reasons, neutrophils are the predominant cells in the first few days of inflammation process. Okay. Now, following the migration of the neutrophils, what all cells will come? So it is monocytes. Monocytes will come. And these monocytes, they can fuse together, right? So 4 or 5 monocytes or even more than that, they will fuse together and differentiate to form macrophages. So these macrophages, again, I'll show you in the graph, they stay at the site of the injury for a very long period of time. Why? Because they have a very long life, unlike neutrophils. Now, the migration of the monocytes, it will continue for days to weeks. And as it is happening, more and more macrophages is also getting formed. But depending on the injury and the implant, it will determine the concentration of the chemotherapeutic factors that is to be released for the monocytes to migrate and as well as like facilitate their further differentiation into macrophages. So this is the graph I was talking about. So this is a graph showing the cell number. Okay, so here intensity basically refers to the cell number or cell density of different type of cell population that we already know, or the different types of leukocytes, and how that intensity is varying over a period of time. So as I mentioned, initially just within the few days of injury, when the inflammation has started, so there, it is the neutrophils that arrive first because the cumulative factors responsible for the migration of the neutrophils that are secreted at the beginning of the injury. Okay, which is why neutrophils are coming, and after like a few days or months, like after a few time, where they are very short-lived, which is why um, their numbers starts declining. Okay, so within two days, okay, so the cells one cell will start to die, and you will see there's a gradual fall in the cell number within say a few hours maybe. Okay, so what I said, so maybe few days, not few hours, within few days. And as its number is declining, we need other leukocytes, right? We need other leukocytes to finish the job if any infectious agent has entered. So in that case, you will see gradually the number of cells like macrophages, their number is increasing. And as I told you, macrophages are very long-lived, which is why after a certain period of time, see here the neutrophil population has declined. But at this point, if you just draw a line, and you will see that at this point, at the same time point, the number of macrophages has reached the highest intensity, and that intensity is remaining constant throughout until the wound is healed. Then, so these are the two important leukocytes. Other than that, the mononuclear leukocytes. So, if you remember our previous discussions, where we discussed various kind of leukocytes like the granular, then agranular, and the, depending on how many nucleus the cell has. So depending on that differentiation, so we can basically say that neutrophils are polymorphonucleosides, sorry, polymorpholeukocytes. Okay. And the other leukocytes which has only one nucleus, so they can be called as mononuclear nucleosides. So mononuclear leukocytes. So you can see their intensity is also the highest at the initial, not as high as the neutrophils, but their number is like not diminishing over the period of time. It's kind of s- slightly decreasing, but not very rapidly increasing. And you can see even in a long period of time, their number stays almost stable, right? Their intensity remains almost stable. So the neutrophils, macrophages, and other leukocytes, like the mononuclear leukocytes, again, the distinction is clear to you, I hope. Other than that, there will be other cells as well, okay, that are needed for the formation of granulation tissue. I'll come to it. So what are the cells you need? You need the fibroblast, okay? Then the blood vessels will start forming due to the proliferation of endothelial cells. So that will also begin. So these two events are long-term events. After inflammation has happened, or you can say after the chronic inflammation has happened, uh, like in acute and chronic, uh, it is classified based on time duration as well as which cell population participates. Okay. So after the entire inflammation process has completed, then the granulation tissue will start forming. And here, these two cells, the endothelial cells and the fibroblasts, they will do the main job, which is why their intensity has increased and has become stable until granulation tissue has formed. Simultaneously, if our implant is of very big size, okay, and it cannot be phagocytosed by the macrophages, then they will start fusing with each other and create something called foreign body giant cells. So there also, when you monitor this process for a very long period of time, you'll see towards the end, these cells, the foreign body giant cells, their number is also at their highest. And lastly, after granulation tissue has formed, then foreign body reaction is happening at the biomaterial interface. Then our body will try to contain it, okay? Because these giant cells, they secrete various kind of hydrolytic enzymes, which is not uh, good for the neighboring tissues, normal cells as well. It can kill them. So our body tries to wall it off, and then the fibrosis will happen. Okay, so fibrosis will again come to it. What is fibrosis? So this is like a image, okay, to explain to you how the sequence proceeds over a period of time. The sequence of events which I explained in the first slide, how they proceed over a long period of time, starting from the inflammation point, not before that. So just quickly, we'll go through these points. The size, shape, chemical, and physical properties of biomaterial, they may be responsible for the variation in the intensity and the duration of inflammation or wound healing process. Yeah, seems easy, right? The intensity or duration of the inflammatory reaction may characterize the biocompatibility of the material. So, if the intensity of the cell of the inflammatory reaction it is very intense, like very high, then then our implant is not biocompatible. Similarly, the inflammation process it goes on a very long period of time, let's say for years, all right? Then also our material is not biocompatible. So while injuries initiate the inflammatory response, the chemicals which are released from the plasma cells or injured tissue, they will mediate the inflammatory response. So we are again going back to the slide where we discussed the formation of provision matrix. So there, the formation of provision matrix happened through what? Okay, it happened through first deposition of the plasma protein and then the cells, right? The leukocytes, the blood cells, they have come, right? And they started secreting chemicals and because of these chemicals, the inflammatory response has started. Okay, and they will also mediate, okay, how the inflammation sequence should happen, okay, how long the acute inflammation should happen, how long the chronic inflammation will take place, or whether chronic inflammation needed or not, okay. So this mediation is happening through the uh, cells interact in the provisional matrix. A different mediator systems they will interact, okay, and they will provide a series of checks and balances so that none of the activities gets out of control. Okay, everything is in regulation. Chemical mediators they are quickly inactivated as well as destroyed, suggesting that their action is local. Okay, predominantly local. It is at the implant site, so they are not allowed to migrate elsewhere because they our body wants to concentrate on the whatever agent that has entered, only on that, not on other part of the body. So it's a local inflammation always happen locally. Remember that. So generally, the lysosomal proteases Uh, so I'm back. Sorry for the disturbance. I think some internet connection, like the internet connection may not be very good here. So as I was saying, the lysosomal proteases and the free radicals. Okay, so these are the most harmful factors which can cause so they are released for damaging that particular implant. Okay, our body consider it as foreign, so it wants to go, want it to go. It cannot really recognize it, so it gives a response which is general to anything foreign that comes to our body. But if they're secreted at a very high concentration, it can also damage the surrounding host tissue, which is still healthy. Okay, that is why this statement is written here. So these are the list of chemical mediators, basically chemicals which mediate the inflammation process, and like their examples. So when I talked about free radicals, I meant superoxide anions, then H2O2 are released. Then under proteases, there will be collagenase, elastase. So these are the enzymes meant for degrading the collagen and the elastin fibers. Then there will be vasoactive factors. So basically, these agents, they will change the flow of the blood, right? Something like that, like histamine. So I think we discussed this in the previous, uh, not previous. I think when we're discussing uh, briefly the immune system or immunology, there we discussed histamine. Other than that, the protease, plasma proteins like the kinin system, then the coagulation or the complement system. So these all like the growth, then some growth factors also are important here, like PDGF. So you just go through this list, you will understand, or if you have ever come across any of these names and you have used and you have seen their usage elsewhere, you will be able to better understand what these chemicals are doing in this course of action. But just I wanted to bring to your attention that these are the chemical mediators that mediate the inflammation process. Now, acute inflammation, as the name suggests, acute means small, so it lasts for a short duration. Okay, and its main characteristic is, or the acute inflammation is characterized by exudation of fluid and the plasma proteins. So there will be accumulation of this fluid and causes the condition called edema. Again, we discussed what is edema in our brief introduction class. Then after accumulation of this fluid or edema, there will be also migration or e-migration of leukocytes. So in the beginning, which leukocyte is predominant? It is the neutrophils. Okay. So neutrophils, as it has multi-lobed nuclei, that is why it is also called polymorpholeukocytes. These PMNs and other leukocytes, they will migrate from the blood vessels to the implant site. And their migration is assisted by adhesion molecules and in part by chemotaxis. So we discussed this, how they migrate. So go back to that again, same introduction class where I discussed the inflammation process. So, and come back if you don't understand these two lines. Okay, and ask if you have any queries. I'll be happy to take maybe in a doubt clearing session. Okay, or you can WhatsApp me as well. So it's not an issue. Now, following localization of leukocytes at the injury site. So once the leukocytes has come, then the process of phagocytosis will begin, which is the process where the cell tries to engulf. the entirety of dead cells. Okay, in this case, the cell will try to engulf the implant. So that process will happen, and for that, the cell will try to secrete some enzymes like protease, etc., to break off or neutralize that. Okay, and so that it becomes, its cell tries to chop it off in smaller pieces, so it becomes easy for the cell to phagocytose. Okay, that seems logical, I hope. And then, so phagocytosis is basically achieved by cells like neutrophils and macrophages. The major role of neutrophils is to phagocytose microorganisms and foreign materials, right? So, right. The phagocytosis, again, we have discussed the steps in phagocytosis, but once again, for the completeness of this topic, recognition. The first step is recognition that the thing that is inside our body is foreign. Some recognition will happen. Then neutrophil will attach to its surface and will try to engulf and degrade it. So remember, when I talk about recognition, I don't specify specific recognition. Okay, there will be non-specific recognition because these cells are part of the innate immune system. Okay, if you have still uh, doubt in this part, just let me know again in the doubt clearing session. I'll try my best to clarify. Then, so first recognition, the neutrophils will attach to its surface, okay, and will try to engulf it. And once it engulfs it, it will try to degrade or kill it. So the later part will not be applicable, or the later two steps, that is engulfment and the degradation, they may not be applicable if the biomaterial is very large in size. Recognition and how, right? So this point makes things clear. Recognition, how recognition and attachment of neutrophils is happening. It is happening through serum factors, which is called opsonins. Okay, so these opsonins, they are basically IgG, which is part of the humoral immunity. IgG is basically immunoglobulin G, and the complement protein, that is C3b. So these two components create something called opsonins, and these opsonins are present in our serum. These opsonins help in recognition part. So re- neutrophils, they are themselves are not recognizing. They are taking help of opsonins for recognition. And then once it recognizes, when the neutrophils are like told that this is foreign, they will be able to attach to it and do the further steps. Now, since most of the implant that we use for any purpose in tissue engineering, those are usually larger in size than the size of the cell. So the cell will try to phagocytose, but it will fail to do it, which is why frustrated phagocytosis phenomena is seen. Because the cell is not able to phagocytose it, no matter how much enzyme it is trying to release. Okay, so it fails, which is why the term frustrated phagocytosis is given. The amount of enzyme release, it will depend on the size of our implant. The larger the size, the larger the amount of the enzyme released. But if the bio implant or the biomaterial that we are using, if it is of a size which can be phagocytosed, like for example, it's a powder or a particulate, very small particulate, maybe in micron size or nano sizes, then it will provoke a slightly different uh, inflammatory response. Okay, then this frustrated phagocytosis will not happen. So the PMNs, that is the neutrophils, they are the predominant cells in the acute inflammation step or the stage, and their numbers or their intensity is like can be seen very high for only a week. I was confused in the previous slide, no? How much for how long neutrophils will stay. So this point makes it clear. So these neutrophils, they will stay only for a week. from the start of the inflammation. And for a week only, we will see that peak. After that, their cell number will start declining. But if they last for a longer period, like more than a week, maybe a month, then we should understand that the cells are not able to do their job because some infection, which is very, very difficult for these cells to clear out, it is present there. Okay. And then the mm, next part is chronic inflammation. So for this video, I will stop here and I'll continue from here the chronic inflammation in the next recording. Thank you. Do watch the next video for the continuation of this topic. Thank you.